Awesome. Thank you for the introduction. Good morning, everybody. I'm excited to tell you about the work that my group is starting. I am interested in engineering the size and shape of plants because in plants, like in all biological systems, size and shape are important determinants of function. And if we can figure out how to engineer these features, we can create some new and really useful plant varieties. On this slide, I'm showing two examples in which humans have already changed the shapes of plants to produce something useful. On the top is modern maize, which was bred from its wild ancestor teosinte to increase both the size and the number of seeds that the plant produces. These changes in maize have made it into one of the most productive food crops we have today. And on the bottom is a dwarf rice plant, which helped usher in the green revolution by allowing farmers to apply massive amounts of fertilizer to their fields and get an increase in grain yield instead of just more straw biomass. Now, what's exciting about these two examples is that their changes in structure are both the result of changes in DNA sequence. And as a genetic engineer, I see this link between sequence and structure as an opportunity to apply some advanced genetic engineering techniques towards changing biological shape. But I'm not just interested in engineering structure in order to make a larger maize variety, although that would be exciting. I'm actually interested in engineering structure because of how important it is for environmental stress tolerance. The shape and angle of a plant's leaves can affect photosynthetic efficiency and the ability of a plant to tolerate heat stress. The shape of its root system determines its ability to acquire water and other essential nutrients from the soil. And structures like these air channels that you can see in these wetland plant stems can be essential for survival in flooded soils. So if the goal is to be able to engineer structure, the question becomes, how do you do it? Well, what we know is that structure requires very precise spatial control over gene expression. And I'm going to illustrate that point by showing you three different Arabidopsis plants grown on agar plates so you can see the shape of their root systems. This first wild type Arabidopsis has a long primary root that grows down in the direction of gravity. It has several lateral root branches that also grow down in the direction of gravity. And if you were to zoom in on this root system, you'd see that the roots are covered in tiny root hairs, which are structures that are really important for water and nutrient acquisition. Now, if you make a single base pair change in this plant that disrupts the amino acid sequence of a developmental regulator called IAA14, you can make several different changes to the plant simultaneously. This plant no longer produces lateral roots. It has a hard time sensing gravity, which you can tell because the primary root doesn't grow straight down. The primary root's also shorter. It's missing these root hairs, and there's several structural changes in the shoot as well. Now, one of the reasons that this change affects so many different structures simultaneously is because the developmental regulator, when expressed from its native promoter, is expressed in many different cell types throughout the plant. Now, if we take this developmental regulator and we instead express it from a promoter that's only active in one cell type, we can make a more targeted change in root structure. Here, the plant does not produce any lateral roots, which is the targeted change we were trying to make, that's otherwise identical to the wild type plant on the left. So our strategy is to engineer root structure using very precise spatial control over gene expression. Now, if you want precise spatial control over gene expression, you have a couple of options. You can go into the literature and you can look for a published promoter that has an expression pattern that you would like to use for your engineering project. If you work in Arabidopsis roots like I do, you're lucky because there are many published promoters whose expression patterns have been pretty well characterized. But if you can't find a promoter in the literature that has an expression pattern that you want to use, you might think that you could use an RNA-seq data set in order to identify genes that are preferentially expressed in specific tissues, and then use the promoters for those genes to drive expression of the genes that you would like to be able to express instead. This approach is actually a little more challenging than it might appear. And this challenge is nicely demonstrated by a paper in which the authors wanted to express this yellow fluorescent protein from the ARF7 promoter. This promoter drives expression of an ARF7 gene, which we know is expressed in many of the cell types in the root tip of Arabidopsis. But if you take just the 5' genomic sequence upstream of the ARF7 gene and you fuse it to the fluorescent protein, you find no expression in the root tip of Arabidopsis. If you go back and instead grab not just the 5' genomic sequence, but the first intron and exon as well, you can recover the expression pattern in the root tip that you were expecting. It tells us that there's information in this first intron and exon region that's important for driving expression of ARF7 in these tissues. And as an engineer, that complicates things for me because I don't know exactly which regions from the genome to pull out in order to get expression of my gene in the tissues that I want. So if you don't have a published promoter that has the expression pattern you want, and it might be difficult to mine an RNA-seq data set, what can you do? What I think is that you can use some logic gates in order to generate new spatial patterns of gene expression. The way this would work is illustrated with these cartoons. Here we're showing the root tip of Arabidopsis with a fake fluorescent protein being driven by this promoter A. It's on in the root cap of Arabidopsis. Promoter B is on in a subset of root cap cells called the columella and in some central vasculature cells. If you combine promoter A with promoter B using a logical AND gate, you should get expression only where promoter A and promoter B are on or in the columella cells. So using this AND gate, you can take a couple of published expression patterns or known expression patterns and generate new patterns. Okay, so in order to so this is our goal. In order to start building logic gates, they first generated some basic building blocks for controlling gene expression in plants. These are synthetic transcriptional activators composed of a DNA binding protein fused to an activation domain and a nuclear localization signal. When they're expressed in plant cells, they should recruit RNA polymerase and initiate transcription of downstream genes. Built using the same set of DNA binding proteins, a collection of synthetic repressors that bind to the DNA but have no activation domain. When they bind the DNA in a region that would otherwise recruit RNA polymerase, they should interfere with the assembly of the polymerase machinery and the DNA and prevent the transcription of any downstream genes. So these are the basic tools that we started building in order to construct logic gates in plants. Just going to show you how you might use them in order to build a logic gate, right? You have these synthetic transcriptional activators and repressors. To build something like the AND gate that I just showed you, you might want to have a synthetic promoter that has binding sites for multiple synthetic transcription factors in it. In this case, our AND gate is going to drive expression of GFP. So our output gene GFP should be on only when we have both our inputs present in the plant cell. In the no input case that I'm showing here, there's a constitutively expressed repressor that's keeping expression of our GFP gene off. Our first input is an activator that would turn on expression of our GFP gene, except that that repressor is still there keeping GFP off. Our second input is a repressor that turns off the first repressor, but because there's no activator in the system, GFP is still off. So it's only when you have both input A and input B present in the cell that should get expression of GFP. We test to see whether or not these things work using transient expression assays in Nicotiana. In this case, we infiltrate 4 different agrobacterium strains into leaves containing combinations of plasmids encoding pieces of our logic gates. So in this first spot, we deliver an agrobacterium strain that only has the output plasmid in it. Our second spot, we deliver a strain that also has our input A transcription factor. In the third, we have the output plasmid and input B. And in the last, we have both of our inputs plus the output plasmid. What we can see is that you get strong expression of GFP only when both of your inputs are delivered to the plant cell. And we can quantify expression by taking leaf punches from these Nicotiana leaves and putting the little discs into a plate reader and measuring fluorescence. Now we can build not just AND gates, but actually using these types of tools, we can design and build all 16 different 2-input logic gates. So far, we've been able to actually construct and test 14 out of the 16 possible 2-input logic gates and show that at least in Nicotiana leaves, we're able to build logic gates of a pretty diverse variety using these collections of synthetic transcription factors. Now, performing logic in leaves is obviously not getting us that much closer to controlling spatial patterns of gene expression in a whole plant. So we started taking tissue-specific promoters in Arabidopsis and using them to drive expression of our input transcription factors in order to do computation in Arabidopsis roots. So we have 2 tissue-specific promoters that we're using as the inputs to these circuits. One is the sombrero promoter that's on in the root cap of Arabidopsis, and the other is this pin-4 promoter, which is on in the columella cells and in the central vasculature, like the cartoons I showed you earlier. Now, if you were to combine these 2 different tissue-specific promoters using a variety of logic gates, you would expect to get a variety of different tissue-specific expression patterns. And when we implemented these logic gates in Arabidopsis, again, by driving expression of the synthetic transcription factors using these two tissue-specific promoters, we can indeed recover expression patterns that at least qualitatively match the expected expression patterns when we are trying to implement these logic gates. I'm going to go into depth on one of these logic gates. This is a really simple gate called a buffer gate. Buffer gates should just recapitulate the expression pattern of the input promoter. So here, if we compare our buffer gate to the input promoter, which is the sombrero promoter, we see that it matches the expression pattern exactly. The way this gate works is that we have the sombrero promoter driving expression of an activator that turns on a synthetic promoter, which in turn expresses the reporter gene. Now, why would you want this? Well, what we've found is that if we change the number of binding sites for the synthetic transcription factor in the output promoter, we can change the amount of reporter gene that's expressed. So what we think is that in a tissue-specific manner, imagine swapping out now this input promoter for your tissue-specific promoter, we might be able to tune gene expression. And when thinking about tuning gene expression, I started thinking about these plants that have no lateral roots, because producing no lateral roots is actually not all that useful to a plant. These plants do really poorly when you put them into soil. If you tug on their shoots at all, they just come flying right out. They need some lateral roots in order to be able to acquire water and to anchor themselves into the soil. And so instead of just completely eliminating lateral roots, we wanted to see if we could apply this ability to tune gene expression in plants in order to tune the number of lateral roots. So we thought is that, okay, if we express a lot of this synthetic transcription factor in that xylem pole paracycle cell in this very specific cell type in plants, we get no lateral roots. But if we turn it down, maybe we'll get a gradient in root branch density. Alternatively, maybe this type of tuning will actually just identify a threshold above which we are completely eliminating lateral root branching, but below which we get essentially wild type levels of root branching. Either way, we wanted to apply these tools to figure out what we would get and what kind of impact on development we could have. Okay, so we designed synthetic systems that look like this, where we have our tissue-specific promoter driving expression of the activator and a variety of synthetic promoters driving expression of that mutant developmental regulator. And when we did this, what we found was actually that even at our lowest number of binding sites for the synthetic transcription factor, we got no lateral root branches. We weren't able to tune down gene expression far enough in order to be able to get any variation in phenotype. The lucky thing about using the bacterial DNA binding proteins that we used in order to drive expression of, in order to build out these synthetic tools, was that we have a really good idea of what DNA sequence these transcription factors like to bind to. So we know what bases we might be able to mutate in that binding site in order to weaken these promoters even further. That allowed us to build a really small but targeted library of synthetic promoters that have, instead of the consensus operator sequence, mutant operator sequences. And these mutant synthetic promoters drove less and less expression of our reporter gene in response to the same amount of synthetic transcription factor. Okay, so when we go back and we apply these mutant buffer gates, we can start to get actually this gradient in lateral root branching. So by expressing this developmental regulator at what we think is different levels inside of the plant, we can get a different number of root branchings. We're starting to be able to tune this phenotype. So that's an exciting way that we, we did some other measurements of root phenotypes to make sure that the impact that we were having was specific to root branching, and that things like primary root length or the ability of the root to sense gravity were the same. So we're only affecting root branching. Okay, so that's an application of one of the simpler logic gates that we built. I wanted to also highlight this not implies gate because it's one of my favorites. Not implies gates are interesting because they are only on when one of your inputs is present in the system, right? As soon as you have both inputs, the gate should shut back off. What that allows you to do is potentially take one tissue specific promoter pattern and subtract out another to refine your promoter or to refine your expression pattern relative to your first input. Now, why would you want to be able to do that? I thought it would be interesting to see if we could tune gravity sensing in roots. So the way that gravity sensing works in Arabidopsis, or at least the current models, are that columella cells are full of starch-filled granules, and that when the root is reoriented with respect to gravity, these granules fall and they tug on actin filaments within the cell, and the tugging on those releases auxin into the neighboring cell types, and it does so asymmetrically. So when you get a large amount of auxin along the bottom of the root, that tells the cells to stop elongating, and the relatively low amount of auxin on the upper side of the root allows those cells to continue, and that asymmetric growth causes a bend in the root that reorients it with respect to gravity. That's a lot, right? But what we were thinking is, okay, can we express genes in just these kind of neighboring cells that are transporting auxin to potentially prohibit transport of auxin and tune the plant's ability to respond to gravity? And so far, what we've been able to do is show that a wild type plant, which grows down in the direction of gravity, has in a confocal image an organization that looks like this, does differently than a plant where we express the same developmental regulator that makes the cells insensitive to auxin. If we express that from a promoter that's active in all of the root cap cells, we get a plant that's agravitropic. This one's actually going completely upward on plates. If you were to zoom in on this root, though, you'd find that the development of these root cap cells is really messed up. They basically form this little tumor-like ball. And if you look at a reporter of hormone signaling, of auxin signaling in the root cap, you'll see that there's obviously an issue with proper development here. Now, if you limit expression of this developmental regulator to these cells along the side, you can still get a plant that's agravitropic, but you're starting to recover a more normal looking developmental signature in the root cap. Now, we don't know for sure whether the development here is, or whether the gate is completely off in these columella cells, because you can see that the root cap organization is still a little bit different than wild type. And so we're going back and trying to re-engineer these to make sure that it's really off in these cells and that the agravitropic signal that you see is not due to maybe a little bit of leaky expression of this developmental regulator in these cell types. But I wanted to highlight this to show that I'm excited about using these tools to start to refine expression patterns and get at more nuanced questions in terms of how these plants are developing, and our ability to try to control them hopefully will become better as we develop more and more of these synthetic tools. Okay, because this is for interest of DOE, we're also interested in moving some of these circuits into bioenergy grasses. Everything I've showed you today so far has been in Arabidopsis and in Nicotiana benthamiana leaves. Now, one of the reasons we were able to push this project forward over the course of my postdoc actually was because we were able to test the activity of our synthetic transcription factors really quickly in these leaf-based transient expression assays. We don't know whether or not the same transcription factors will be functional in grasses, right? Monocots are relatively different in terms of expression requirements than dicots. And so we are trying to develop protoplast assays in the lab in order to really quickly test to see whether or not these same types of these same types of synthetic transcriptional regulators will be functional in grass cell types. So this is just pictures from my lab where we're starting to use protoplast for transient expression. And so far, we've been able to test one of our bacterial DNA binding proteins fused to several different synthetic activators. These are the same type of synthetic transcription factors that we've been using in Ceteria. And what we found is actually that one of the synthetic transcription factor designs that worked really well in Arabidopsis is our weakest one when we move into Ceteria. And so we're interested in continuing to test some of these tools in these transient expression assays so that we can then have a better idea of how well these circuits will work when we transform them into grasses in stable lines, because we know that that's going to take a lot of time. Okay, so as we move forward, lab is interested in applying some of these approaches into other plant species, and also in engineering bacteria, and then testing the engineered plants and bacteria that we've made for environmental robustness and their effect over developmental time. Okay, with that, I just want to say thank you to Jose Deneni, who was my postdoctoral advisor, where I did the majority of the work I just talked about, and to the people who are helping me build out my lab at Stanford, and also to all of you for your attention. Thank you.
Get us started with a quick question. So when you repeat the motif sequence in there to kind of have the stronger output, do you ever see any issues with silencing?
Yeah, so we don't see issues with silencing with the repeated sequence at the promoters. When we built original, like some of the early designs for these, I was reusing terminator sequences and reusing the 35S promoter in some of these designs, and those were very quickly silenced. But the gates that have just repeated DNA binding sequences, we've been able to assay out into the T3 generation, and they still don't show any signs of silencing.
Cool, great,
Yeah.
super interesting. Yeah.
Yeah.
Any other questions from the audience here?
I have a question here. I'm curious about the two logical circuits that you haven't been able to validate yet. Are those particularly tough? Is it specific to that system, or just a type of circuit that is tricky in general?
Yeah, so those are exclusive OR and exclusive NOR gates, and they are notoriously difficult to build. We've designed them, but they require, at least with the type of tool we have, 9 different synthetic transcription factors, all functioning properly together in the cell, which is more than our largest gate that we have working. So I think just with that type of complexity, we haven't been able to tune it.